Hereby I open this academic ceremony in which Sarah Kochs will defend the academic thesis Focus on Food, Effects of Mindset, Hunger and Dietary Restraint on Attention Bias, Food Intake and Brain Responses to Food. Welcome to the audience. Before we start the defense, may I invite you to present a summary of your study and the conclusions of your thesis.
Thank you. Dear Prorector, dear members of the Defense Committee, dear audience. Today, I will defend my thesis entitled Focus on Food, Effects of Mindset, Hunger and Dietary Restraint on Attention Bias, Food Intake and Brain Responses to Food. First, I will give a brief overview of the research I conducted for this thesis. I will start with presenting the background of my research, that is, today, we live in an obesogenic environment. This means that we are surrounded by a vast overabundance of extremely tasty, high-caloric food. This environment encourages the overconsumption of food. As a result, many people are overweight or obese and face associated negative health effects, such as cardiovascular diseases and diabetes. In addition, our current society embraces thin bodies as its beauty ideal, creating a conflicting food environment. On the one hand, overconsumption of highly tasty, high-caloric food is encouraged through the temptation arising by its omnipresence. On the other hand, a thin body shape is encouraged by society's beauty ideal as well as by favorable health outcomes of a healthy body weight. For some people, more than for others, maintaining a healthy body weight is difficult in this environment. In face of this obesogenic environment and societal norms, many people try to control their body weight with dieting, meaning that they consciously restrict their food intake. This often manifests in dietary restraint, which describes the intentional restriction of food intake motivated by body weight and shape concerns. Yet, restrained eaters often have a higher body mass index than unrestrained eaters. Therefore, it is important to understand the eating behavior and motivation of restrained eaters. Two potential research avenues on dietary restraint have been the investigation of brain responses to food and the investigation of attention bias for food. Attention bias for food refers to preferential attentional processing of food stimuli over other stimuli. In both domains, the evidence is very inconsistent, with some studies finding differences in brain responses to food or attention bias for food between restrained and unrestrained eaters, while other studies find no such differences. Dominant theories in this research field propose that brain responses to food always reflect the rewarding properties of food, and that the reward value of food is always higher for restrained eaters than for unrestrained eaters. Therefore, it is commonly assumed that research participants always focus on hedonic aspects of food, that is, taste and pleasure obtained from consumption. But this might not be true. Especially tasty high-caloric foods, which were mostly used as stimuli in previous studies, are ambiguous. They are highly tasty and therefore highly rewarding, so they have a high hedonic value. At the same time, they are associated with weight gain and negative health effects, so they have a low health value. This ambiguity means that people are likely to fluctuate in their view on high-caloric food. At times, they may focus more on hedonic aspects, like comfort obtained from eating, while at other times, they may focus more on health-related aspects of food, such as negative effects on body weight. This means that people will fluctuate between a hedonic and health mindset when thinking about tasty, high-caloric food. Restrained eaters who are concerned with their eating behavior and body weight may especially fluctuate between these hedonic and health perspectives. These perspectives will be referred to as attentional focus or mindset, and both terms will be used synonymously during the remainder of the presentation. Now let's have a look at the research conducted in this thesis. We investigated effects of current states, mindset and hunger in particular, and dietary restraint. We looked at how these factors affect brain responses to food, attention bias for food, and food intake. And we expected that the outcome measures would be most pronounced in a hedonic mindset and that effects of mindset would be strongest in people with high levels of dietary restraint. To test our expectations, we conducted 3 empirical studies that are presented in chapters 2 to 4 of this thesis. In chapter 2, we used fMRI to measure brain responses to food and investigated the effects of attentional focus and dietary restraint on these responses. In chapter 3, we measured eye movements and manual response latencies as measures of attention bias for food and looked at the effects that mindset and dietary restraint have on it. In chapter 4, we measured manual response latencies as measure of attention bias for food and looked at the effects that hunger and dietary restraint have on it. In both chapters 3 and 4, we conducted a mock taste test to measure food intake to assess the effect that mindset or hunger together with dietary restraint have on food intake. In this presentation, I will focus on chapter 2 and present the methodology and results in more detail, and I will only give a brief summary of chapter 3 and 4. I will start with this brief summary. Basically, in chapter 3 and 4, our expectations were not confirmed. We observed no effects of mindset, hunger, or dietary restraint on attention bias for food. We also did not observe any effects of mindset on food intake and also only negligible effects of dietary restraint. Now let's have a more detailed look at chapter 2. In this chapter, we presented pictures of food to our participants and measured their brain responses to these images. While presenting the food pictures, we controlled the attentional focus of our participants with a one-back task. In this task, food images were presented in quick succession and the participant had to compare the food item currently presented on the screen with the previously presented one. We induced a hedonic attentional focus by asking participants to compare food items based on their taste. To create a health attentional focus, we asked the participants to compare the food items based on their calorie content. As control condition, we asked the participants to compare the food items based on their color. So by manipulating the attentional focus, we can control the mental processes that the participants engaged in while viewing the food pictures and did not have to rely on assumptions and inferences about these processes like most previous studies did. We tailored our food stimuli to the individual taste preferences of each participant. In this way, we presented personalized, highly tasty and highly untasty, high caloric and low caloric food pictures to our participants. On the slide in the background, you can see an example of how food stimuli could have been categorized. It is important to note that this selection looked different for each participant as it was based on personal taste preferences. Each participant also rated the tastiness of the chosen food items on a 10-point scale, with 1 indicating that a food item is absolutely not tasty and 10 indicating that a food item is highly tasty. As you can see by the numbers presented on the slide, tasty food items were indeed rated as being very tasty with an average rating of approximately 9, and untasty food items were indeed rated as being quite untasty with an average rating of approximately 3. So tasty and untasty food stimuli used in the study were truly perceived as highly differing in tastiness by our participants. We analyzed our data using 2 analysis approaches. We used univariate fMRI analysis, which is the standard approach to fMRI data analysis, as well as multivoxel pattern analysis. In univariate fMRI analysis, the average level of activity in a brain region is considered, while in multivoxel pattern analysis, the pattern of activity in a brain region is considered. This distinction is important because these analyses might be sensitive to different types of information that is present in the fMRI signal. It could be the case that 2 stimuli, like the pizza and the hot dog shown here, elicit the same level of brain activity, but elicit very different activity patterns. Therefore, the distinction between those 2 stimuli that is present in the fMRI signal would be overlooked when analyzing the level of brain activity, but could be detected when considering the pattern of brain activity. We expected that effects of attentional focus together with dietary restraint would determine brain responses to food when considering the level of brain activity. We expected that brain regions associated with reward processing would show the highest activity level in the hedonic attentional focus, while brain regions associated with conflict and control would show the highest level of brain activity in the health attentional focus. We expected that differences in the level of brain activity evoked by attentional focus would be strongest in restrained eaters. We expected that food characteristics, that is tastiness and calorie content, of the food would be reflected in patterns of brain activity. We observed that the level of brain activity was not influenced by the tastiness of food and barely by its calorie content. Unexpectedly, we also observed that dietary restraint did not influence the level of brain activity. But as we predicted, we observed that the level of brain activity indeed was influenced by attentional focus. Several brain regions, many of them belonging to the reward system, showed the highest activity level in the hedonic attentional focus. So how can these results be interpreted? The level of brain activity did not differ between highly tasty and highly untasty food stimuli, while these stimuli strongly differ in reward value. This means that the level of brain activity in response to food cannot reflect its reward value, as this would require observing differences in activity level between tasty and untasty food, which we did not observe. So this observation goes against the dominant theory, which poses that increased brain activity in response to food reflects increased reward value of food. Instead, tasty and untasty food do not differ in salience. They are both highly salient, especially in a hedonic attentional focus in which the taste of food is central. So the level of brain activity possibly reflects the motivational salience of food instead of its reward value. This fits with the observation that many brain regions showed the highest activity level in a hedonic attentional focus, in which the taste of food is highly salient. When we looked at the patterns of brain activity, we found that information about tastiness as well as about calorie content was present in those activity patterns. We found that this information was present in several regions of the brain's reward system, among others. So these results suggest that it is necessary to look at patterns of brain activity to obtain information about how the brain reflects tastiness and calorie content of food. This shows that not only analyzing the level of brain activity, but also analyzing patterns of brain activity will be necessary to fully understand how the brain processes food. So what can we conclude from the current dissertation? Looking at chapter 2, which was just presented, we saw that the level of brain activity did not differ between highly tasty food, which is highly rewarding, and highly untasty food, which is absolutely not rewarding at all. This suggests that the level of brain activity in response to food cannot reflect its reward value, as this would logically require a difference in activity level between tasty and untasty food. Yet, both types of food are highly salient, especially in a hedonic attentional focus. We indeed observed that the level of brain activity was strongly influenced by attentional focus, so it seems likely that the level of brain activity in response to food reflects motivational salience instead of reward value. From the multivoxel pattern analysis results, we can conclude that food characteristics are reflected in patterns of brain activity instead of level of activity. Therefore, both types of analysis will be necessary to fully understand how the brain represents food. Unexpectedly, we observed no effect of dietary restraint on brain responses to food, so it seems that brain responses to food are more influenced by situational factors like attentional focus than by more stable behavioral traits like dietary restraint. From chapters 3 and 4, which were only briefly mentioned during this presentation, we can draw the following conclusions. We did not observe any evidence of attention bias for food in restrained eaters. Considering this, in combination with the inconsistent literature on the topic, it seems that attention bias for food is not the most relevant factor in driving altered food approach behavior in restrained eaters. And surprisingly, we did not observe any effects of mindset on attention bias for food or food intake. So it might be that mindset has less influence on these variables than we expected, or alternatively, certain preconditions might need to be met for mindset manipulation to affect behavior. In previous studies which observed effects of mindset on attention bias for food, the participant had an active role in the mindset manipulation, while mindset was manipulated passively with video clips in the current study. So maybe an active involvement of the participant is necessary to affect behavior. Future research is therefore necessary to figure out the exact role of mindset in attention bias for food and food intake. Thank you very much for your attention. Now I give the word back to the prorector.
Thank you. Thank you for this clear and interesting presentation. We will now start the defense, and the opposition will be opened by Professor Nederkorn. She is professor in cognitive psychology of eating at Maastricht University and also the chair of the assessment committee. Go ahead.
Dear candidate, with great interest I read your thesis on the role of attention, food reward and brain responses and the relation with eating behavior or restrained eating. The thesis showed great care and interest for methodology and how the field can be improved. I really liked the care and for all your experiments, and my compliments for you and also for your supervising team. But I have a question for you. An existing theory proposes that, and I now call them overeaters, but they might be restrained eaters or obese people, but people who tend to overeat, that for them food is more rewarding or more salient, and this could explain their tendency to eat too much. To test this theory, people with and without overeating tendencies were compared in the scanner, while they were freely viewing foods. Their brain responses were measured, and difference were often thought to reflect difference in reward, or the rewarding value of the food. However, as you also now explained, the results are not consistent between studies, and it's also very difficult to know what the activity actually means. Does it really reflect reward, or are there maybe health concerns? And how do you know that a specific pattern in the brain really reflects reward? We don't really know for sure. Therefore, you had a different approach. You manipulated the focus of the people in your experiment, and next you measured their brain responses. That's a smart thing to do. However, it does answer a different question. You know now how brain activity looks when someone thinks of the taste of food or the calories, but you still do not know what a person on the street thinks, so an overeater, when she or he sees food in a free viewing situation. But that was actually the first question that people had. And I thought about your experiment and I thought, well, you did this nice fMRI experiment, so you know how individual responses to hedonic and health aspects look like. So you kind of calibrated their individual responses. Why didn't you add a free viewing period after that? And then, knowing the individual responses, you could know where they were thinking of when they were freely viewing food. So would that be possible? And if yes, why didn't you add such a free viewing period?
Highly esteemed opponent, thank you very much for your compliments and your question. If I understand your question correctly, you are asking why we did not add a free viewing condition to our design, because this free viewing condition would be more resembling of the natural state when people perceive food.
Well, at least without an instruction, so you could see what they more naturally would do.
Yes, indeed. We decided to not add this free viewing condition because of a number of reasons. First, we do believe that the environment in the scanner is not comparable to the environment in a natural food perception situation. So the fact that someone is laying down in the scanner and cannot interact with the food in a way that would be possible in a normal scenario might immediately bias the person towards a different response than would be in a normal food situation. Probably the people in the scanner also feel watched. I mean, I guess they will think we look into their brains, so they might have more negative attitudes toward high-caloric food in that situation just because of a social desirability effect. And on the other hand, if we would add this free viewing condition and then would observe some brain activity in response to that condition, how would we know how we could interpret the brain activity that we observe if we do not know what the mental process of the participant is in that way, in that condition?
Yeah, because you calibrated that in your first part of the experiment in which you did have the manipulations. So you have kind of a comparison material.
I'm not sure if you can compare the conditions that we used previously with the condition that would occur in the free viewing condition, because we only manipulated 3 mindsets basically, but there are other things that people can think of, and they might even think of it differently than they did in the exact way we phrased our mindsets. So I would doubt that that is really comparable.
Okay, thank you very much. I find your arguments very sound. But then I would think that to answer the question what people think of food when they are in real life can actually not be answered in an fMRI scanner. Because they're always lying down in this feeling watched. All your arguments are always true when you are in an fMRI scanner. So actually what people normally would think of food in real life cannot be tested then ever with an fMRI study.
I think there's some truth to this, but I think we can achieve an approximation with the situation in the fMRI scanner, because we try, with our mindset manipulation, we try to simulate certain situations or thoughts that people might have about the food, and I think in this way we know the mental process that happens when the participant looks at the food picture, so we know how to interpret the brain activity, but in this way we kind of simulate as good as we can how the person perceives the food, because I do think that also in real life, outside the scanner, people will likely have their own mindset and their viewpoint on food.
Yes, I agree, but you still don't know if in real life, without your manipulation, they have this mindset. So you don't know if overeaters have this mindset more often than non-overeaters, because you kind of instructed them, but in real life, what they do, you don't know.
That's true, and that would be a very interesting study to assess what mindset overeaters do have in real life, and if there maybe is a difference in sort of a chronic mindset that overeaters engage in, whereas people who have no problem with overeating do not engage in this so often.
Can I add a very small question? Yeah. In your presentation now you said that unexpectedly you did not find differences between restrained and unrestrained eaters. This kind of adds to my point. I wouldn't expect differences because of this, because you ask people to look at the rewarding value or of how the taste, or on calorie, and you do this in everyone. So why would you expect a difference of restraint? Because you kind of remove that part by instructing the participant.
That's a good question. Indeed, the manipulation that we used appears to be very strong and maybe kind of simulates what restraint does in a natural environment. So maybe the effect of restraint really is just composed out of being in a hedonic viewpoint more often than in a health viewpoint, or having more fluctuations between these two different perspectives.
Thank you very much for your answer. I'm very happy and give the word back.
Thank you. The opposition will be continued by Professor de Jong. He is Professor of Experimental Psychopathology at the University of Groningen.
Thank you. And dear candidate, I also read your thesis with great interest. En in Groningen, u spreekt ook Nederlands, hè? In Groningen zeggen nou, het was bepaald geen straf om het te moeten lezen. So it was really nice. But it has also a bit of a negative side effect. And actually, because reading so thoroughly about food, about all kinds of tasks related to bogus taste tasks, hedonic, for whatever reason, the health mindset did not occur that much to me, and especially for those who are a bit difficulty with inhibiting their impulses, it was well, anyway, it was a nice thesis, and what I especially liked about your thesis as well was the very thorough introduction of why the current status is a bit disparate. So there are so many different types of findings, and it's difficult if you write an introduction, for example, an article about attentional bias and restrained eating or eating more generally, to give a proper coverage of all the things that have been done. And then you conclude that, yeah, actually, one major explanation for why we have this very difficulty in making sense of all the data is that these people use all kinds of different paradigms and all kinds of different parameters within these paradigms, different ways of analyzing these data, and also to do not take into consideration all kinds of things like the health mindset or hunger or other motivational states. So, and then you started your research. And then in the end, and I only saw that after reading your thesis, also your propositions, and I would like to start with a question about Proposition 9 in that regard. In Proposition 9 you say, obesity research would benefit from the use of more standardized procedures, such as standardized paradigms and analysis approaches, to increase comparability and reduce inconsistency of findings. And of course I fully agree, I think. Most of us will agree. And I was just wondering, when you look back to your own contribution to the literature, how you appreciate then your own work in retrospect with regard to this proposition. Did you add to the consistency, or, well, how do you consider your own research, for example, in study 2 and study 3 in that regard?
Highly esteemed opponent, thank you very much for your compliments and your question. So, if I understand the question correctly, you want to hear my estimation about how I see how my research adds to increased consistency in the research field that my thesis falls into. Yeah,
apparently you give a kind of a guideline for all of us to be more consistent, and I just wondered, hey, how did you comply yourself to this particular guideline?
Yes, I do think that by using a task-based manipulation, this will help adding consistency to the literature, because by having a very clear idea of, for example, how brain responses to food have to be interpreted, by knowing what the participants were thinking about, we do take out a lot of noise that has been present in previous studies that contributed to the field with passive viewing, where there's, of course, always the problem of reverse inference, meaning that a brain region can have several functions. Mm-hmm. And of course, we don't know which of the functions it is carrying out at the moment when the people look at the pictures of the food. I think in this way, this thesis does add to increased consistency in the literature, or gives an attempt to, let's say, improve the field a bit. And also with the studies that we did, we did not use in all studies the same tasks, but we did try to make approaches to improve the tasks as much as possible, to give as clear results as possible. And also with the mindset manipulation, where there I wanted to take out some variability between participants in the way that they look at food. And I think the main approach or idea is by taking out this variability, by making it very clear in which way they should look at the food, in which condition.
Yeah, I can see that. But still, if I was reading your thesis and I just read it from A to Z, and then I had that logical order, and then you did a study with a singleton task, and I thought initially, hey, this is really nice also to have this task-irrelevant feature there, and very clever to use that type of task for this particular aim. But then you didn't find any attentional bias, also no consistent effect of the manipulation on the attentional bias, also no individual differences with regard to restraint. And you came with some speculations why that would be the case. First of all, you say, well, perhaps it was too easy. There were too many trials where this distractor popped up, for example. So you have all kinds of, but perhaps also you could say, hey, it's a new task. Perhaps this particular task was not suited for measuring attentional bias. So I expected then for the next study that you would then use again the singleton task and improve it a bit, and then have a, but then you selected a completely different task, a visual search. And so this, I was just wondering, hey, but why this break? Why not continuing in the singleton, or why not go back to the literature and say, hey, there are a couple of studies that actually showed, for example, the impact of hunger on attentional bias, or did show differential effect on restrained eaters, for example, with the rapid serial visual presentation paradigm. where also the food item is task irrelevant. Hey, and then continue with manipulation rather than doing again another type of task. So not only that you do another task within your own series of studies, but also related to the available evidence in the literature. So I was just wondering how you see that.
Yes, indeed. It would have been probably a better choice to go on and improve the additional singleton paradigm for a third study, but this unfortunately did not work out due to practical reasons and time constraints.
Okay. So if you would have the opportunity to continue there, you would, or let's say to your colleagues like me, to continue with the singleton task and to see whether you could continue and find results. Is that what you're saying?
Yes, I think that would be a very interesting research avenue. I'm not sure how promising it will be, but I think it would have been indeed an improvement to the current thesis to stick with a more consistent line of paradigms.
Okay, thanks. Do we have an opportunity for 1 minute? Yeah, and it is just also, I was also struck by your presentation when you put these pictures in my face with all these nice, tasty food items. And in the introduction, you make a very strong case, and you were very outspoken, that for a proper attentional bias, this ecological validity, it should be something that is task irrelevant to measure the proper bias, because people should be very immersed in a task, and then even fully immersed in the task would be distracted, so a kind of a vigilant or engagement bias for the food items. But then if you present me with this obesogenic environment, then there is food everywhere, and also, if I speak for myself, although I'm often very dedicated to my task, I also have lots of times that I was just mind wandering, or I just walking, or I look in a magazine, or I walk in the street and have a superficial chat. So I doubt whether it's just always the case that it's most sensitive in real life, the attentional bias most important, when you are so involved in the task. Why would not be these other times of life where you just do something but not very much dedicated, also very important? So that's another question related to the importance of the singleton task. Why is it so important that it should be task irrelevant?
I do think that it is most important to study how distraction by food works in situations when people are different with another task, busy with something else. And then food intrudes, because I do think that these are the most problematic eating moments when mindless snacking will occur, which will very likely contribute to an energy surplus, whereas in situations where people do have more the deliberate intention to consume food, that is probably more controlled eating that will take place, and probably can of course also lead to weight gain, but is less likely to lead to excessive weight gain that could become problematic and even lead into obesity. I do think that this mindless eating, eating when distracted, eating when stressed, is the most problematic eating behavior in our society.
Okay. Well, thanks very much for your further explanation. Then I give the word back to Dietary.
Yeah, thank you. We will switch over now to our online opponent. That is Dr. Papies. She's reader in the psychology and of behavior change at the Center for Methods and Meta-Science at the University of Glasgow. Go ahead, please.
Thank you. And let me start my question today by giving congratulations to you and to your supervisors on an impressive piece of work, Sarah. Your thesis presents a really, I found, sophisticated and carefully justified set of experiments with lots of different methods and lots of different approaches on a quite complicated question. So a lot of previous research has attempted to shed light on the relationship between this construct of restrained eating and responses to food cues to help us understand a bit more about those people who continuously try to restrain their food intake. So they have been called today also overeaters, but they might also be restrained eaters. So already that tells us they're a bit complicated. So there's a lot of literature here and a lot of different things to navigate, which I felt you did really well in your thesis. So because you're clearly the experts on this notion of restraint, I'd like to ask you about that to get a better understanding of this for myself, and also to hear your thoughts about how to best proceed with this research. So I'm wondering, for this construct of restraint, what is it really? So you write that it refers to attempts to reduce food intake to control your body weight because of these body weight and body shape concerns. So that almost sounds like it's a behavior, or it's attempts at a behavior. So I'm interested to learn what are the key kind of psychological features of this construct. So what really drives the psychological trait or characteristic of restraints?
Esteemed opponent, thank you very much for your compliments and your question. So if I understand your question correctly, you are asking me to further define and specify what I understand with dietary restraint. I do think that dietary restraint is a very difficult to summarize and classifiable construct, because I do think even around this construct there is a lot of debate or controversy about the literature, what is really meant by a restrained eater. You do have the side that defines it as a person that is indeed attempting this behavior, having the attitude about him or herself as a dieter, does these attempts, but is most often unsuccessful, and there is a cycle of restricting and under-eating, basically. And then there is the other definition of dietary restraint, which more looks at successful dieters that, more act out on their behavior consistently. In this thesis, I used the first version of the construct, so looked more at these restricting, breaking the restraint, cycling dieters, because I do think that this is the type of restraint that leads mostly to problematic behavior, which could have negative health effects and which could lead to obesity. Therefore, we focused here on the definition and used in all of the studies the restraint scale by Herman and Polivy to define the construct.
Okay, cool, that makes sense. But indeed, if I remember correctly, this restraint scale from Herman and Polivy is quite old. It's a scale from the 1980s. And I was wondering, for you coming into this field as a young and ambitious researcher, how would you ideally measure restraint if you now have the opportunity to design a new tool from scratch to measure dietary restraint in the way that you just described it? How would you approach this? What kind of questions would you ask to your participants?
That is indeed a very difficult question. I think if I would go about to design a new restraint scale, I guess I first would start conducting surveys to see what people who identify themselves as dieters. So I would start out with having the question, do you see yourself as dieter? And then I would try to find out more about the characteristics that these people have. And then, probably with factor analysis, would try to figure out which factors would best describe the population that I'm interested in. But going on from this point, I am not so sure if there is even maybe one type of dieter, or if you maybe would need to have different scales to identify different types of dieters. I imagine that there are many different types of personalities which might affect how one approaches dieting, and then in the end how dieting affects behavior. And I also think that the motivation that persons have with respect to dieting will have a large influence on how they approach it. And I think I would, up from the data, try to figure out what these views and different types would be.
Yeah, I think that's a great approach. It really would be a more data-driven approach in a way than maybe a theory-driven approach, because it seems a little as if the theory of restrained eating by Herman and Polivy really kind of shaped how the construct was measured. And I guess related to that, because you talk a little bit about context effects also in the introduction of your dissertation, the context for the original development of that theory and of the scale was North America in the 1980s, or maybe even late 70s and 80s. And I guess probably the food environment, so the context was very different from the Netherlands and also very different from now. So do you think that dieting is different now, 40 years after this scale and the theory was developed? And does this have implications for how one should study it, or for the cognitive processes that shape the dieting behavior?
I do think that dieting, in a way, has changed compared to 40 years now, alone by looking at what you can see on the internet, on social media about dieting trends. I do think back then there was a certain culture around it, but this culture now with the new media has changed a lot, and probably the way people approach it, the social aspect of it has become probably even more important than 40 years ago. And yeah, I do think, therefore, probably also the way how it's measured could be, could be renewed. And probably this internet media culture in a way also has diversified it a bit, but streamlined it a bit more on other areas. And I do think that this, of course, could not be taken into account 40 years ago.
Yeah, that's a really interesting point. I hadn't really considered the kind of the social media and the social context. I had more thought about the food environment, but really both have changed a lot over the past decade. So indeed, I think that's really good to take into account. I guess related to something that you brought up earlier, and this I think would be the final bit for my question for just now, is that indeed people can have different kinds of goals, right? You refer to different kinds of reasons for dieting, but people can also have non-dieting reasons for restricting their food intake, right? And so I'm thinking about climate change in this specific context. So people are increasingly concerned about the climate impact of their food. For example, when you choose a dish in a restaurant, you may now forego that beef burger because of its huge carbon footprint, or other people may use oat milk instead of cow's milk in order to reduce the greenhouse gas emissions associated with dairy production, which are immense. So I'm wondering, is that similar to a kind of dieting restraint that you discuss in your thesis, but with a different long-term goal? And do you think that this kind of regulation of one's eating with a different long-term goal is implemented through similar cognitive and neural mechanisms, or are they different, and why?
Brief answer, please.
Okay. I would think that the motivation why someone diets will affect the way how they respond, how they go about their sort of dietary restraint. And I would think that it is a sort of diet, of course, if you, for example, restrict your intake of animal products. But I would say this is a different type of dieter than people who do it merely for appearance reasons or people who do it for health reasons.
Great, thank you so much.
Thank you. The opposition will now be continued by Professor Goebel, Professor for Cognitive Neuroscience at Maastricht University.
Thank you, Prorector. Dear candidate, first want to congratulate you to this really nice thesis, which I read with great pleasure, and also congratulations to your supervisors. I liked it very much that you had an active task in the Chapter 2 fMRI study, and also that you analyzed it both for activation levels as well as multivariately. This was very nice to see. I have a few questions, and the first one is actually to your choice to binarize the stimuli in extremes, right? You have palatable food, high palatable food and low, and the same for the calorie content. And I like a lot your conclusion that maybe you find something that they are not differentiable because they're both, for example, palatability, are both highly salient, similarly highly salient. And then I thought, wouldn't it be then not smarter to not binarize, but to keep also mid-level, so the whole range basically, use a covariate analysis, And then you might disentangle that because you would have also items which were not salient or not that strongly salient. And you could then prove more that your hypothesizing is correct, right? Now it's because you have this extreme and assume they are leading to similar results, you can actually not verify that independent or with your own data, you know, because it could be anything else. What do you think about that?
Highly esteemed opponent, thank you very much for your compliments and your question. Indeed, this is a very interesting perspective. And I think it would be really interesting to test this out with a more varied set of food stimuli indeed varying as well as the whole spectrum of tastiness, as well as the whole spectrum of calorie content, and to see if then the activity, the palatability or calorie content would be observable from the level of brain activity, or if still only the patterns would reflect those characteristics.
Exactly, yes. Then I want to continue with both the results of the activation study and the MBPA results. First, to the activation studies. So if you look at your figure 2, which you also had in your presentation, the attentional focus results look very nice, but they, in my mind, when I first looked just at the picture, not even reading anything, I thought, wow, she found some very nice networks, brain networks, right? Like deform mode or saliency or attentional network. So did you consider, because you have this precuneus activity, you have this medial frontal activity, you have this lateral on the junction of temporal parietal lobe activity, and these are typical hubs in these networks I just mentioned. So did you consider that could also be akin to some of these networks, what you could, for example, do to compare the mass of these networks with your results and see to which networks they load or overlap maximally? Did you consider that?
That is a very interesting approach. Until now, we did not consider comparing the activation patterns to these networks.
Okay, because it could validate also your idea about the saliency, for example, if it loads on the saliency network.
Yes.
Yeah. Another question I have to the table 4, when you show the ROI results, and you show nicely also here the beta values there, which I really like. It's very transparent, very detailed. One can look at many interesting aspects. One thing which jumped in my eye is that you find also brain regions which have, of course, the right order of beta values. If you look, for example, for the strict nice result that you have highly palatable food, attentional focus, and then health, taste, health, and neutral or color, then you find nicely the order of the betas as you have looked for. But I also see that, for example, for the first entry, but there are others too, for precuneus cingulate gyrus, you find negative betas quite strongly, right? So you have the lowest beta is -3, and the highest is 0.04, so basically 0. So how do you explain that you have strong negative beta values?
To be honest, I don't have an explanation for this.
Okay. So actually, I asked this question with the purpose of relating back to these networks, because many of these networks, for example, the deform mode network, are usually high, because you think about yourself. And then if you do a task, like your attentional focus task, you attend to something and the networks go down. And that could explain such negative effects in those regions which belong to these networks. Of course, this is now difficult. What I'm saying is speculation. It could be another explanation. But it would be nice to look at these networks because you could really tease a bit more out of your results in that way. Another question actually is related. Sorry, I looked very nicely at your data. I really liked it very much. So I turn now to figure 4, which is the nice results of the MBPA, right? And there you could decode nicely caloric and palatability. But again, when I looked at that, I was surprised that the highest accuracy values, which are actually quite low, we have to be clear here, right? They are not super high. But if you look at the stronger one of these ones, they are actually in the visual cortex. You see actually in the posterior visual cortex where the calcarine sulcus is, very yellowish colors. You see them on the lateral side of the visual cortex, very yellowish colors. So the stronger effects are in the visual cortex. Of course, you find other regions. And in your analysis, you actually did a smart approach by saying, I look in those regions which have been previously reported, right, and show what your effects are. But if I would look independently of previous results, just now from what I see here, just look at your figure, I would come to the conclusion, oh, that is nice. Caloric content as well as palatability are mainly in the visual cortex coded, you know, and not in, you know, these prefrontal and orbitofrontal other areas or mesocorticolimbic regions, as you call them. But I would say the strongest effects are in the visual cortex. What do you answer to this kind of question?
Indeed, it is noticeable that there's quite some strong activation in the visual cortex in our multivariate analysis, but I do think that this is not uncommon. This is not the only food-related study that does find activation in the visual cortex. So that is, and it might also be that it does have to do with the visual processing of food, because, I mean, we did just the decoding analysis, so we don't know actually what the classifier built the distinction on.
Mm-hmm. Could you also, this is a very good answer, could you also think about something which has to do with connectivity?
Maybe it could be connectivity.
In a more specific sense, right? Information goes up the visual hierarchy, it goes also down the visual hierarchy, right?
Indeed, yes. There could be, of course, some processing going back to the visual areas from the reward centers.
Exactly. So we could have nicely top-down effects which boost the visual patterns which you have in the visual cortex, right? So this could be a feedback effect. And that brings me to a point, as methodologically nice your study was, you could even add, even on pattern basis, effective connectivity analysis. So you could really look, how do some areas in some parts of the brain connect, speak to, effectively influence, directionally even if you want, but at least correlation-wise, look how these areas and even patterns speak to each other, explain variants somewhere else in the brain. And that would be another dimension which adds nicely to your activity, patterns. Pattern connectivity would be the next thing you could also try, which might answer some of these points. Therefore, I mention it, right? So they could reveal maybe very novel things if you look how these areas speak to each other and explain each other, bottom up, top down, or even simply on correlation basis. Yeah. Do I have still time? Okay, then I thank you very much. Thank you very much.
Thank you.
Give it back to the corrector.
The opposition will be continued by Dr. Smul, Assistant Professor at the Department of Cognitive Neuroscience.
Yes, thank you. First of all, I want to express my admiration for the thesis. My question will be about chapter 2, and in this chapter, I think you really did a great job with the MRI analysis, like Professor Goebel already mentioned, the GLM, then the Bayesian analysis, and MVPA-based decoding. So it's really wonderful. And that in over 50 participants, which I think is a tremendous job. But my question is about proposition 10. So let me read it. How the brain reacts to food is not a fixed characteristic of a person, but depends on attentional focus. So interventions may target that attentional focus to influence how the brain reacts to food. And now in chapter 2, you manipulated attentional focus in quite a simple way by having people focus on taste or on calories or on the color of stimuli in a one big task. And now I wonder, if it is that easy to manipulate attentional focus, well then, those changes in focus last. So does it make sense to target such a flexible and changeable variable mental process as attentional focus in an intervention? Now, I'm not a clinical psychologist, but I would guess that in therapy you would optimally want to make changes in some system that is relatively fixed, but of course not so much fixed that it cannot be changed in the first place. So what do you think about it? Do you think that it would be possible to make lasting changes in attentional focus, and if so, how?
Esteemed opponent, thank you very much for your question and your compliments. if I understand your question correctly, you're asking about if I think how that it would make sense to manipulate mindset in order to affect behavior, for example, in the context of therapy.
Yeah, if it's so flexible, it would easily change back after therapy.
Indeed, I do think that there are different conceptualizations of mindset, and in chapter 2 we used a very flexible conceptualization of mindset, therefore we also called it attentional focus, and I do think this attentional focus can switch in an instant from being on health and then seeing something changing to hedonic. That is indeed true, and I do think that it was a good approximation to study it in the fMRI as a first attempt with this easy to manipulate strategy. But if we would want to translate these findings into behavior change programs, for example, I do think that a more substantial, longer lasting conceptualization of mindset would need to be chosen to do it.
Yeah, I can imagine. Okay, thank you. I have another question, actually about reaction time analysis in chapter 2, because you did the fMRI analysis in very advanced and cool ways, I think. But the reaction time analysis, I think you did something strange with the outlier deletion. So you deleted outliers at level 1, so a single trial level, which is a normal thing to do because it generates more stable reaction times. But for that you used a criterion which was defined across participants. So the mean and the standard deviation across participants. So that would be a level 2 parameter. So in a way you're mixing up level 1 and level 2 effects. So I think what you do by that is by using this cutoff, if a participant is relatively slow, it loses more of its slow trial. So you make the slow participant on average faster, and the fast participants they are being cut off on the left of the distribution. So you make them a bit slower. So you're reducing individual differences, which sounds as a strange thing to me, because if you do your statistics at level 2, you want to estimate those true individual differences and not reduce them. So is this a new method, or?
Well, maybe indeed it would have been better to stay at level 1 for the reduction of the outliers. Since we used this analysis of reaction times only as a measure of accuracy, we did not pay too much attention to this, I think.
I see, yeah. Well, anyway, it probably didn't have that much of an effect, because you're right that you only lost 1.23% of fast trials and 0% of slow trials, which actually is an indication to me that there's something strange happening, because usually the outlier reaction times have a positive skewness, or a tail at the right. So there are typically outliers would be slow responses. So those you want to get rid of, and fast responses are far less common. Whereas in your data set it was the other way around. So yeah, I would recommend to try to do it on single participant level. If you have enough trials, things like that, it also counts. Yeah. So I don't think it was things went very wrong, because you did essentially nothing or hardly anything about outliers. Yeah. Okay, that was my question. Thank you.
Thank you very much.
Yeah.
We'll have a little bit of time for an extra question. May I request you?
Yes, I do like to have an extra question. You looked in your analysis at 2 aspects, or in your studies. the hedonic value of food, but also the caloric content. But as a previous our guest from Glasgow also said, there can also be other ways to look at food. So, for instance, the price, but also where it comes from, whether it's organic or whether it's animal-based. And I also thought about the time. That kind of refers back to the previous question, how fast people switch between different concepts. And maybe that's really fast, or maybe people even have two aspects at the same time in mind. So why do you think, and maybe they are switching faster than a bold response can exist. So how do you think about that? Why did you choose these two aspects and not more? And why do you think that you have either one or the other focus at one point of time?
Highly esteemed opponent, thank you very much for your question. We chose these two foci as example of foci because we think that these are very common ones that most people will have, and also to keep the design simple. But it would be interesting to see how and if other foci would affect brain activity. And it might be the case that
Sarah Kochs, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense. I suggest that you and your company await the results of our deliberation and our return in this room.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Southside, 7 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. All your time, 'cause I'll go the extra mile. Long road to the south side, 7 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Hit the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. Even though these nights seem long, I know that I am never on my own. Know you've won the battle once you've grown. Southside, 7 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. Long road to the south side, 7 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side right to the west side. No place like home. If it's questions that you've got, go the extra mile and die. What's on the line? I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. 'Cause I'll go the extra mile.
Sarah Kochs, the degree committee here present has assessed the quality of your thesis and your defense. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Roefs is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to take the floor.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
Okay. By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Sarah Kochs, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with a degree certificate signed by the rector, the secretary, and the other members of the committee and affixed with the official seal of the university. Dear Sarah, my laudation is entitled, How Much Evidence is Enough? Today you became Dr. Kochs, for which you deserve huge congratulations. I sincerely hope that you are very proud of yourself now. I, for one, am very proud of you. I'm very glad that you're here today as Dr. Sarah Kochs with a beautiful PhD thesis. As most of you know, a scientist's job is to develop a theory, a testable hypothesis, and then critically test this theory. Evidence can then either support or refute a theory, and if a lot of evidence is found in support of a theory, one can have some faith that this theory is good, that it's true. Today, I would like to present all my evidence in support of the theory, Sarah is a true and good scientist, which I collected during your PhD project. And then in the end, you can all, and you as well, Sarah, of course, draw your conclusions based on the evidence that I present. So let me take you back to when it all started. At the end of 2016, my colleague Shan and I were searching for 2 PhD students on my VD project on neural representations of food. And the ideal candidate needed to have a strong neuroscience background and expertise in eating behavior research. We received many applications of candidates who either had a strong neuroscience background or expertise in eating behavior research. You were one of the few candidates who had it both. You completed a research master in cognitive neuroscience and had a strong interest and expertise in eating behavior. So as the first piece of evidence, Sarah had all the necessary skills and background knowledge to embark on this scientific project. During the 2 interviews we had with you, Shan and I asked you many questions. And both of us are not really known for asking easy questions. And we were impressed by how you handled the questions and also politely disagreed with us when necessary. We had some good discussions. And that is, of course, what we look for in a PhD student and a very important characteristic of a scientist, someone who can think for themselves. So here's the second piece of evidence. For the research of your PhD thesis, you collaborated closely with our colleague, Leo Pimpini, your parent here today, and you proved to be an excellent team, with each of you having their own strengths. You collaborated for 5 years, and I was impressed by how well you worked together, supporting each other and truly valuing each other's expertise. So here's the third piece of evidence. You're good at team science. For each of your 3 projects, as well as those of Leo, a lot of coding was needed. You already had a strong coding background, and you developed that further and collaborated with Leo. You wrote well-designed scripts for running the experiments as well as running the analysis. You tested your scripts very thoroughly, but you would still ask me if I were certain that the scripts did not contain any mistakes. Of course, one can never be sure about the absence of mistakes, and you had to learn how to deal with that bit of uncertainty, which you did. We all benefited greatly from your admirable coding skills, and new PhD students will certainly be able to build on these scripts. So here's the fourth piece of evidence. You have an essential skill for a neuroscientist. Very good coding skills and a great eye for detail. A good scientist dares to challenge the status quo, even if everyone in the field seems to believe it, and when something makes a lot of intuitive sense. Let's call it a convenient theory. In your thesis, you challenged the dominant theory in the field that the reward value of food is reflected in the level of activity in the so-called brain reward system. So a stronger brain response in the reward system if we see food that we like than if we see food that we don't like. So here's the fifth piece of evidence. You think for yourself and you challenged a dominant theory in the field supported by some famous scientists. Your research convincingly showed that when tested in a well-powered study and properly analyzed, there's no evidence for this dominant theory. Using Bayesian statistics, you showed that there was overwhelming evidence for the null hypothesis, which means no difference in brain activity between very tasty versus not tasty foods. So here's the sixth piece of evidence. You design and analyze your studies very well. Throughout these 6 years, you showed real stamina. You never gave up, worked incredibly hard, and even in the face of setbacks, such as technical struggles, a cyber hack, COVID, housing problems, journal rejections, unfair editors. I've seen you grow over these years, not only as a scientist, but also as a person. So here's my seventh piece of evidence. You worked very hard and patiently and never gave up. And with good results. Your large eye-tracking study on the effects of mindset and dietary restraint on attentional bias for food was published in Journal of Cognition. Moreover, a few weeks ago, Neuroimage invited you to submit a revision of your manuscript on the effects of attentional focus and dietary restraint on neurorepresentations of food. I've hardly ever seen such doable reviews. So another publication is coming up soon. And this is already my eighth piece of evidence. A good scientist is not easily convinced, and one piece of evidence is often not enough to convince a good scientist. Sarah is definitely not easily convinced, which is the ninth piece of evidence that she is a true and good scientist. So over these years, I've collected 9 pieces of evidence supporting my theory that Sarah is a true and good scientist, and non-refuting this theory. So what would you conclude now? And what would you conclude, Sarah? You can also tell me later. I would say that this is really abundant support for the theory. And I sincerely hope that this amount of evidence also convinces you, Sarah, Dr. Kochs. I hope you enjoyed this afternoon and that you will have fond memories of today. I would also like to congratulate your parents and your partner, who can be very proud of their Dr. Kochs. Enjoy this special day together.
Dear Dr. Kochs, also on behalf of Maastricht University, I would like to congratulate you with this performance. I think you did a wonderful job and you can really be proud of yourself. I would also like to thank your supervisors and your family and friends. And I would also like to take the opportunity to thank all opponents for being here and for the contribution, especially those from far away, Glasgow and Groningen. Thank you very much. And before I end this ceremony, I would request you to stay here a bit for pictures taken. And the audience, you can already go to the reception with some drinks and possibly some foods. And enjoy the rest of the day. Thank you.
Dankjewel.
Ja.
Redelijk.
Ja, nou, de mensen in de zaal waren heel tevreden.
Hartstikke goed.
Bedankt. In de besprekingen waren allemaal heel tevreden dat je het echt super goed hebt gedaan. Ja,
Dat is leuk om te
alleen
horen.
maar complimenten